Okay. See if we can get things going. Okay, so my goal today is to convince you guys that helping the heart relax is actually going to improve its performance, which is what the focus of my research is on. So I think everyone here is familiar with the idea of the job of a heart is to pump blood through the circulatory system, deliver oxygen and nutrients to your entire body. And your heart is a muscle. And the muscle cells that power your heart are these cardiomyocytes, which are these rod-shaped cells. You can see we can isolate these in a petri dish, and what you'll see is that they'll beat, they'll contract, and they'll relax, which mimics the heartbeat in the lab. And these are specialized cells in that you're basically born with the same number of cardiomyocytes that you have throughout your whole life, which means these cells are really programmed to live 100 years, which if they do that, they beat over 3 billion times. And so what's fascinating about these cells is we're all sitting here, hopefully at rest, our heart's pumping about 5 liters a minute. But a trained athlete during exercise can pump up to 35 liters a minute. This is a huge dynamic range, and it's these cells that are responsible for adapting to that stress. And so we're familiar with what happens to muscles when they have to work harder, i.e. are under stress. They grow, and that process is called hypertrophy here. And so a classic case of hypertrophy would be Arnold Schwarzenegger right here exhibiting hypertrophy, the skeletal muscle. So your heart is a muscle, it does the same thing. So I was at the University of Pennsylvania. Here's 2 pictures of hearts that we had for research of human hearts. You can see a normal-sized human heart, and at roughly the same scale, a hypertrophied human heart. Now, I can't tell you just by looking at this whether that's a diseased heart or whether that's from an athlete, right? And the reality of it is that you get the same sort of visual result from all these different types of stress. So whether it's exercise or whether it's hypertension, where your heart has to pump against an increased pressure, an increased load, it leads to the same type of growth. And these cells that are doing this are expanding and growing because, like I said, you have the same number of cells, which means for that heart to be 4 times bigger, those cells have to become 4 times larger. So that's a massive undertaking for these cells to be able to do this. And while I said I can't tell by looking at it, if I pick up this heart, I can tell which one is diseased or which one is an athlete's heart because the stiffness is different. So there's something about the molecules that are in there that are changing, and this is what we want to understand as researchers. So if you just look at the leading causes of death by risk factor in the world, the top 3 out of 5 are hypertension, obesity, and diabetes. And so not surprisingly, because we just listed that these are cardiovascular stresses, the number 1 cause of death in the world is cardiovascular disease. It outpaces cancer by about 2-fold. The same thing is true here in the United States. About 1 in 4 deaths is due to cardiovascular disease, but this really isn't the entire picture. If you look at just these risk factors, we're all adults, about half of us have hypertension. About a third of us are obese. So all of us are undergoing this pathological remodeling right here in this room, effectively. And so this is a big problem, and we want to understand what those molecular pathways are. So hypertension, obesity, and diabetes typically come together. And as clinicians, we know that this leads to a certain type of heart failure. And so that's this HFpEF, or heart failure with preserved ejection fraction. And like the name suggests, preserved ejection fraction, the contractile behavior of this heart's normal. What happens to these hearts is the muscle becomes stiff, and that impairs its ability to fill with blood and to relax. And so these stiff hearts pump less blood, not because they can't squeeze, but because they can't fill. And so we're very interested in my lab in understanding what is the molecular signature of this stiffening, how is this changing, and then can we reverse it? And so when we think about stiffness, what defines our shape, right? Cells have different shapes. Our shape is defined by our skeleton. So your cells have effectively a skeleton too. And the bones of your cells are called the cytoskeleton, this family of these filamentous proteins. So actin, which forms a thin filament of your muscle, is loaded in these cells, whether they're diseased or they're normal. So here's an example of that, right? And this has been studied pretty heavily. But until we really started our research, people hadn't really looked at these other filaments as candidates for this. And so if we image the microtubules inside a cell, you could see these stiff tubes run longitudinally along the contractile axis of the cell. So the obvious question is, well, what are these things doing when the cell has to shorten? I mean, shouldn't they be oriented in a different way? And so we can do this with advanced microscopy in the lab. So here's a video of those microtubules. And you could see, hopefully, when these cell contracts, things buckle like springs. So the idea here is that these are resisting the contraction of the heart. And we did just sort of a back of the envelope calculation based on how many are here and how stiff they are. And theoretically, this really should affect the contraction and relaxation of the heart. So then the next question we had was, well, what about in disease? And so we had the supply of human hearts at Penn. And if you look on the side over there, failing human cardiomyocytes have this increase in the density of the microtubule network. So there's this 2 to 3-fold increase in these stiff tubes. These cells are stiffer and they have this increase of microtubules compared to non-failing heart cells. So the big question is, well, how does this affect function? And that's sort of the question that we're trying to answer here and how it's dependent on etiology of heart disease. And so in the lab, what we've developed are these thin sheets of myocardium. So we could use a biopsy sample for something like this. And the idea is that we have these things working in a way that mimics the contraction of the heart. And so now we can try different drugs on this and figure out what's going to happen with different patient populations. And so for the microtubule question, of course, we're going to use this drug colchicine, which eliminates the microtubules from the tissue. And amazingly, what we found was that when we depolymerize the microtubule network with colchicine, we see that the colchicine softens the failing heart tissue. And the effect that this has is this actually improves the performance of the tissue, and not by helping it contract, but by helping it relax faster and stretch out more. And so we're really excited about this. And the next steps would be to figure out what the pathways are, what different types of stress drive microtubule network remodeling, and how, and if we can reverse this. So, I mean, of course, this is a great place to be, and I couldn't do all this work without all these amazing colleagues. And then specifically, this working to relax was coined by Emily Hancock, who is right here. And this was actually at the CVRI dinner, where I think it was I'm going to forget the name of it, but where we had to do these elevator pitches. So thank you very much for your attention, and that's it.